Hi everyone, my name is Associate Professor Jessica Mar and I'm from the University of Queensland. I'm really excited today to be able to present some of the career highlights of my research, and I think my career can be summarized by the title here, Modeling the Variability of Gene Expression to Understand How Cells Work. So as a bioinformatician, what we spend a lot of time doing is trying to understand regulatory networks. And these are really important because we live in a world now where we understand that genes don't operate in isolation. And in fact, they're regulated and controlled by multiple processes. And what these regulatory networks are important for is driving these cell phenotypes. And phenotypes are behaviors that cells might adopt. So what we spend a lot of time doing is really understanding what are the genes, these ball things over here, that really drive this network, and also what are the connections between these genes that are important for defining a cell state. And obviously as cells transition from one phenotype to another, like for instance an untreated cell that's been exposed then to some sort of cancer treatment, the network here changes, and it's our job to really understand from the data what these connections are and how they're working to change the phenotype. So what's really exciting now is that our sequencing technologies have changed so much, and one thing that's really fascinated me throughout my entire career is this idea that cells are inherently heterogeneous. And what we mean by that is that from cell to cell there are really key differences, especially in the transcriptome. And before, many of our techniques relied on smushing cells together and understanding on average what was happening. But what's really exciting now is that we live in a world where we've got access to single-cell next-generation sequencing technologies. And these are allowing us to really understand how cells are different from each other. So for instance, we can figure out which are the red cells versus the blue cells, whereas before this was really not possible. And because of that, what's really happening now is that all streams of biology are undergoing massive revamping in how we understand transcriptional regulation. So it's a really exciting time right now to be doing science. So why are cells inherently heterogeneous? And that's because when we think about all the different regulatory steps that go into controlling a cell, there's a lot of room for variation. And so as you all probably know, DNA is one of the important regulatory codes out there. And along the stretch of DNA, there can be many different kinds of variations. And some of those variations are structural. So the sequence variations also lead to SNPs or polymorphisms. But DNA alone doesn't define a cell. And we know that DNA then gets transcribed into RNA, which then becomes protein. And along the way, each of these series of information can also have different variations. And the variations have functional consequences, which overall confers robustness and diversity for these cells. But from the data's perspective, it can also make things much more complex to figure out what's going on. Early on in my career, I was really curious about variation in terms of variance. And by this I mean gene expression variance. And the reason why this is exciting is because most of the time when we think about gene expression, we're looking for differences in average. So for instance, you might have a control group and a disease group. And what we do is we prioritize those genes that have large differences in average expression. So you can see one of the statistics that we often use is the T statistic. And as you can see from this formula, what we often rely on here is a large difference in average expression, that's the numerator, and this is balanced by a small degree of variance, which is in the denominator. And so for anyone who is familiar with engineering, this is basically a signal-to-noise ratio. And what it means is that we end up prioritizing those genes that have a large difference in average and a small amount of variation in that signal. And this is a really sensible way to identify what are the key regulators when you've got thousands and thousands of genes. But a scenario that is not reflected here is what happens when there's a difference in gene expression variance. And so below the line you can see some cartoons of how that might happen. You might have one example where there is a difference in average expression, but as you can see in the disease population, there's a much wider level of variance compared to the control. And the other more extreme example is where you actually have no difference in average expression, but you can see that there's very different degrees of variance there for control and disease. So what I really wanted to do when I was a postdoc at Harvard was investigate this question of whether we could actually obtain information by looking at gene expression variants and not just relying on average expression. So a really exciting place where we could test out this idea was through a collaboration with Christine Wells and Alan Mackay-Sim, who was a former Australian of the Year. And at the time, both of these people were based at Griffith University, while I was a postdoc at Harvard working with John Quackenbush. And so what Alan and Christine had developed was a whole patient bank of factory neural stem cells. And what was really exciting here was it gave us the opportunity to study how gene expression was varying from patient to patient in these 3 different groups. So we had access to patients with Parkinson's disease, schizophrenia, and also a matched control for comparison. And so what we did was we used this model to really understand how variance in gene expression could tell us more information about these diseases. And effectively what we did was we took the data, we did a few different comparisons to really understand how variability in gene expression was driving some of the changes we saw due to the disease, and also at the functional level, what this meant for specific genes that were important for the disease, but also in terms of pathways and networks. And essentially, the punchline from this work was that we identified that within a network of genes, there were different regulatory constraints that played out in terms of low variance or high variance. And you can see with this kind of cartoon how genes operate within a cell. We can see that the degree to which they're expressed is important. So some genes are upregulated or downregulated. But also our key finding here was that different degrees of variance were also functionally important. So you can imagine how low variance might indicate greater stability of certain genes, especially if we're looking from cell to cell or from patient to patient. And you can imagine how that has a trickle-down effect for the other genes that gene might be involved in regulating. So for instance, a receptor controls a kinase downstream that might then trigger a transcription factor. And so that sort of increase in variation can also result in greater heterogeneity, more plasticity or noise in this particular branch of the signal transduction pathway. In my career, I've taken these ideas in different directions to build on them and ask more interesting questions. And one example of this is working with a student, Yu Hasagawa, where we wanted to ask what variation or variance in gene expression could tell us about human embryonic development. So here we had access to a data set that was published by Yan et al. In Nature Structural and Molecular Biology. And here we were asking the question of what happens when we look at low amounts of variability from cell to cell within the same embryo versus high degrees of variability in the same embryos as they transition from really early on, so 4-cell stage, all the way up to blastocyst. And we developed some simple statistics to identify how variance was changing. And we found a few really interesting things, namely that as the embryos transition through development, we saw globally an increase in gene expression variability. We also were able to identify key regulators using changes in variance that weren't previously identified before. And these were markers that were really important for each developmental stage. And finally, we found that genes that were very stable, those that had very low degrees of variance, were important for controlling different processes for the cell, especially the embryo as it develops. So these included transcription, translation, and metabolic pathways. So this was a really great example of applying ideas to a previously published data set to understand at a greater detail what was going on with these embryos as they develop. If we think about variance of gene expression, this reflects the width of a distribution. And in statistics, we can represent that distribution through a few different ways. And so we've taken these ideas about variance of gene expression and really started to ask, how does this work with different distributions? So we know that data is often distributed according to a normal distribution. So this is our familiar Gaussian or bell curve. But in statistics, there are actually different kinds of distributions we can use. So this includes the Cauchy, which is similar to normal but with higher tails. We can also have the log normal, which is skewed to one side. Also the Pareto, which is much more extreme in its skewness, and the gamma distribution, which is quite flexible in the different sorts of shapes that it can adopt. And finally, we can think about more hairy kind of distributions, like the bimodal distribution that has two peaks, or by extension, any sort of multimodal distribution. So a while ago, we were really interested in asking, when we think about genes that make up a transcriptome, is it fair to assume that they all follow the same distribution? Because in our bioinformatic models, we tend to make that assumption that one distribution fits all genes. So working with one of my postdocs, Laurence de Tarante, and our programmer Sam, they constructed a classification pipeline to take gene expression data from the Cancer Genome Atlas and basically assign or determine which was the most appropriate distribution for this gene expression profile. And from this work, we found some really interesting conclusions, namely that there's actually a lot of non-normality in the cancer transcriptome. So in the 3 patient cohorts that we looked in, we found that there was over 50% of these genes that were non-normally distributed. So that's quite insightful that our assumption of normality, or of our assumption of a single distribution, doesn't necessarily apply to the whole transcriptome. We also found that when it came to predicting patients' survival, that incorporating the shape of the gene expression distribution actually improved that prediction significantly. So this was really interesting work that really tested whether the assumption of one distribution really fits a transcriptome, and from that so many interesting things were discovered. If we take this idea about distributions a little further, another place where we've had some great success is to ask about bimodality in gene expression data. And so I had a talented MD PhD student, Dr. Daniel PK, who worked with me, and one of his projects was really to investigate the prevalence of bimodal distributions in cancer genome data, specifically breast cancer. And from this work, he developed models which are part of a bioinformatics software package called OncoMix. And essentially what OncoMix does is it identifies oncogene candidates that basically follow this bimodal profile in the cancer patients, but have a unimodal profile in the matched normal. And from this work, we identified several new oncogene candidates for breast cancer data, including one gene that we're really excited by, CBX2. If we follow CBX2 a bit further, what we found was that overexpression of CBX2, which identified this upper peak in the distribution, we found that these patients had much lower rates of survival. And finally, we were excited to see that CBX2 is overexpressed only in healthy human testes tissue. So in terms of following this gene up as a potential drug target for cancer, it's really exciting to see that its overexpression in a normal tissue is of this type for breast cancer patients. So more recently, with my group in Australia, what we're trying to do is bring all these different ideas together and build on this to understand more about how cells are working. And I have a PhD student, Melindri, who's been investigating the question about different distributions in the context of single-cell RNA sequencing data. So here, the similar concepts apply as what we were doing before with Cancer Genome Atlas data, but instead there are different kinds of distributions that we need to use for single-cell data. And partly this is to account for the number of zeros that we see in this particular data, which in general makes it really quite challenging to work with. But what Melindri has done is she's developed a statistical framework to basically decide, based on the profile, which of these distributions is more appropriate. And she's applied this to a host of different data sets, including ones that refer to aging and also Down syndrome. And we're in the middle of finding out some really exciting results from this work. So finally, I wanted to conclude with where my research program is going. And since moving back to Australia, my work has taken on a greater focus towards understanding aging. And what we're doing is bringing the idea of heterogeneity, single-cell RNA sequencing to develop bioinformatic tools and models to really understand how humans age. So I'd like to end my talk by acknowledging all the amazing people that I've had such a great opportunity to work with. I ran my first group at Albert Einstein College of Medicine in New York, and I had a wonderful team there. But I'd also like to thank my group now at the University of Queensland, who've worked really, really hard to set up these projects, and right now we're seeing some exciting results come out. So thank you very much for all your attention, and if you'd like to get in touch, here are my contact details.